Welcome back, everybody. Hope you had a pretty fun break. We're moving on from our car session to episode 4 of the evening. We're going to move into some psych-soc stuff, namely sensation and perception, the cool side of psych and soc, the sciencey side of psych and soc, and we might even do a couple experiments today. Should be pretty fun. So what's going on, Kelsey? Welcome back. I got a lot of the same folks in the room. We also have a couple newcomers. So if anyone is brand new to the game, just to give you a little tour, at the bottom we have the Q&A. Questions in there go directly to myself and all the other teachers. So in fact, we've got Kristen, Chuck, and Dee all in the room. So if you have any questions throughout the entire class, don't hesitate. We got 4 teachers in the room to basically answer any questions we can. Should be pretty fun. And so yeah, we got a lot of the same folks. So welcome back. Also, notice in the bottom right-hand portion of the screen we have a download. I do recommend downloading that. One thing is a heads up about differences, though, is that you won't need to really have it accessible like we did in the CARS class because we're only going to do discrete questions today. So all the information you'll ever need to solve a question will be directly on the screen. We're not going to go through a full passage on sensation and perception. So it's a pretty fun class, though. We get to go through a lot of nitty-gritty details. But I do want to warn you, we could talk about a single one of these concepts for hours and hours, and so we are going to avoid getting overly specific about the nitty-gritty details and mostly focus on general themes, big picture, big takeaways, and the things that AMC does say that they definitely want to test. So I think it'll be fun. I think you guys will enjoy it. Let's have some fun, and welcome to the room. All right, so let's get the party started and talk about sensation and perception. Our main goals today will be distinguishing between sensation and perception. You know, like how, of course, they're part of the same process, but they're different parts of the same thing, so to speak. We'll talk about a couple of special senses, different types of receptors. We'll talk about object recognition, differences between them, and then we'll also talk about, we'll go through all the senses. We'll talk about vision, we'll talk about hearing, we'll talk about touch sensation, olfactory, taste. We're going to go through all the little bits of it. It'll be pretty fun. All right, so the very beginning, of course, notice we're going to focus on the science knowledge. This is very much a content class, heads up. If you do not have any background in sensation and perception, this might move a little quickly. It might be a little bit over your head, but as a heads up, most of the things we discussed today are discussed in Chapter 2 of your Psych-Soc book because that is the chapter that focuses exclusively on sensation and perception. So a little heads up on that. So no worries if it goes a little bit over your head. Feel free. You can always come back and watch this again. We'll really focus on the big takeaways and what matters most. All right, so what we'll start with, of course, is a stimulus. And so a stimulus, of course, hits a receptor. And once it hits a receptor, it goes to a sensory ganglion, which then, of course, goes to the central nervous system, where we will, of course, perceive it. And so now there's a couple different types of receptors. We're going to do very big picture first, and then we'll get more specific in terms of the details. But these are, of course, the most frequently tested receptors. We're going to focus on reception first. So what's a photoreceptor, and where would you find it? We'll get more into the actual pathway soon. We'll get into the brain stuff. Don't worry. We're just going to get big picture first. Yeah, in the eyes. Exactly. Rods and cones. There you go, Bellany. Exactly true. In the retina. Totally true. So photo, of course, photo, receptor, receptor. Hair cells. Now, tell me about hair cells. The ones that we talk about most for humans. I know other animals like cats, yes, they do have the ones that they use a lot. But we mostly talk about the ones in the ear, a little bit nose. Most of the things we're talking about, though, are in the ear themselves. Pili as well, we've got them all over our body, but we do have a lot of specialized hair cells that specifically do work with hearing. Where are those actual hairs inside that help you hear things? Inside the cochlea, exactly. You got it. So we'll go into the specifics of the cochlea. We'll talk about the structure, but talk about the basilar membrane, the organ of Corti. We'll get real specific about that stuff. But also, where do we perceive rotation and linear acceleration? That's actually a rotation movement in the semicircular canalis area. But we'll get into the specifics of those in a bit. They're also where, though? How do you perceive that? In the ears. They're also in the inner ear. There you go. We call them the vestibular senses, I believe is another name for it. It's in the vestibule. You got it. Semicircular canal is right there as well, but that's more of a rotational motion. We'll get into both of those very soon, so we'll discuss those. How about our nociceptors? What are nociceptors? Yeah, pain. You got it. Exactly true. So it's, of course, pain receptors. We do have different types of pain receptors. We'll talk about those when we get to them a little bit later on as well. How about thermoreceptors? This one's a bit more straightforward. Temperature. You got it. So thermotemperature. So those are your temperature receptors. Osmoreceptors? All that. Nice. Concentration. You got it. So it's concentration of the blood. Here's a question for you. Where do you find most of your osmoreceptors? Where in your body would you find most of them? Nice, you got it, Jennifer. It's in the hypothalamus, actually in the hypothalamus, because what does the hypothalamus end up doing? It eventually gets to the kidney belly, that's true, but why would you start? There we go. It produces ADH. Of course, it stores it in the posterior pituitary, and it's also it's the master gland. It maintains homeostasis, right? And so since it's the master gland and we use hormones to mostly maintain our blood osmolarity through, of course, ADH, aldosterone, that's more of a volume thing, but same idea. Our hypothalamus is basically the king dog when it comes to anything endocrine. And we also do, if you haven't been to it yet, we have 2 different channel classes on endocrinology. So we get really into the hypothalamus, pituitary, adrenals, all that stuff in those endo classes. Definitely check them out if you haven't already. How about olfactory? Olfactory is your smell, exactly true. That's the smell receptors. We'll talk about that and how it's actually different than the rest of the senses, interestingly enough. And then finally, taste receptors. Well, that's just taste, as you can see. It's that one's more straightforward. Taste receptors, of course, are taste are on the tongue, exactly. And we'll talk about the 4 different types of flavors and how, yes, when you talk about taste, part of it is olfaction, part of it is taste. We'll talk about kind of the similarities and differences, but there are some significant differences between taste and smell, even though they are very highly associated. And as you probably know, you can actually overcome your taste receptors via smell. Have you guys seen those experiments where they basically have someone bite an onion, but they hold an apple up to their nose and they think that they're eating an apple instead of an onion? You guys seen those experiments? It's kind of funny. Yeah, you can actually totally trick people because it's processed the same. The incoming and the pathway is totally different, so it is quite different. But they've done some funny experiments where you basically make someone smell one thing and have them taste something else, and they get confused and think that they're tasting something different than they are. Yeah, exactly. Do to someone else, as Jennifer says. All right. We'll do some fun experiments today. We won't do anything with onions today, but we will do some experiments on all of us, which should be pretty fun. All right. So let's talk about thresholds first. So thresholds, we got to talk about some kind of a stimulus, which results in sensation. Sensation then results in perception, right? So the perception is becoming aware of it. The sensation is the actual input of the stimuli itself. I'm going through all of our different receptors that we just covered. And so, as we said, this is the raw input, the signal to the central nervous system. The perception is the processing, the perceiving. It's the experience. It's linking to memory, what you've perceived in the past, what your biases are, and all of that stuff as well. So perception can be very biased. So, for example, 30 degrees Fahrenheit might seem warm to someone from the center, the inland empire, as they say, of Alaska in the winter, whereas 30 degrees Fahrenheit is incredibly cold for someone who lives in Southern California. So perception is biased. That's kind of one of the big things there. But Alaska can be nice too. People give it a hard time, but it can be really nice up there. Just not in the middle of winter. All right, so what threshold is threshold refers specifically to the degree that you're going to perceive a difference in perception. And so there is a specific threshold. In fact, there is a very specific person's name describes the threshold. Do you know whose law that is that describes basically what the threshold is for different types of perception? This is actually a law you do want to know. It was also in your diagnostic exam as well. Yes, Weber's law. Nice work, Kristen. Yeah, so Weber's law talks about that. We'll go through a couple examples of Weber's law and how it looks different for different types of stimuli as well. So we'll show some examples. So for example, here's an example of threshold. Can you tell how this is a difference of threshold? The example on the bottom of the page. Photoreceptors, exactly true. Can we discriminate that these are actually 2 totally different colors? They're very similar, but they are different. And so yeah, these are 2 different colors. The one on the right is slightly darker blue, than the one on the left is slightly lighter blue. And so it's like if you can't distinguish the difference, that would mean that this is below your threshold in difference. And we'll talk about the J and D and just noticeable difference on the next page. So that'd be one example. You do color. How about this one? So that'd be saturation or color. Frequency, right, would be color. This would just be size differences. This one's very clearly different between the 2 sizes, and that would be a very noticeable difference in size. All right, and so we already saw this one, of course. This one is an imperceptible difference in size, yet a perceptible difference in color. Even though those are technically slightly different sizes, it's very hard to tell, but the one on the right I think is a tiny bit bigger. Tiny bit, I think. We'd have to look real carefully to see for sure. I agree, yeah, I think it is. I think the one on the right's a tiny bit bigger, but it's meant to be imperceptible, but we're just really good. I was going to say, we're just really good at this. All right, there we go. So this is Weber's law, as we said, perceptible differences. This is the key, though. They're proportional. So I'm going to give you some funny examples of a guy lifting weights. So now, the thing to know about threshold in Weber's law is that you have to set a baseline, and a baseline must be determined for individuals. So we're going to use this guy and his baseline, of course, as our whatever it is. So now I also want to let you know that in order to really talk about Weber's law, you have to have an initial stimulus and a second stimulus, because your data is going to be different if you start with one versus the other. So for all these examples, we're going to say this guy is left-handed, and he's going to pick up the weight on his left hand first. So just as a heads up, this is a left-handed dude, so you know. And also, the reason that that's his left hand and not his right hand is because whenever you see an example of a person or a body, it's always the patient's perspective, right? So just be aware, our right is his left is our right. It's the same every time if you were to, like draw an example of a heart. If this is the heart, this upper part right here would be called what? The part that I just put a circle in. That would be considered your left atria. Yeah, exactly. So this is the left atria. It is everything in science. It's always patient perspective, right? And that'd be left ventricle below it. You got it. So same thing here. We'll use, since we're talking right and left, we might as well tie in some heart anatomy because we're talking about it in the first place. All right. If you have questions on heart structure, atria, ventricles, you want to talk about your mitral valve versus your the tricuspid valve, let us know. We can definitely get into it in the Q&A, but we won't do too much other talking about hearts today. All right, so notice here, so notice this is his threshold. They feel just a little different, so this is going to be defined as this patient's threshold value. So what would you define his threshold value as if we know he's starting with the weight on the left and then picking up the weight on the right? And this is just to the border that he perceives the difference. What would you call that? The J and D for this guy lifting weights, the just noticeable difference. Think about the percent. It's proportional, right? So it's like, you could think percentage differences or like, not 0.6, be careful. So think about, so he picks up a 4-pound weight, then he picks up a 5-pound weight, and he could just barely feel it. So how much heavier is the 5-pound weight than the 4-pound weight in terms of proportions? There we go, Katie, exactly. It is 25% heavier. So of course, this guy, we would say his threshold then is 1/4. Does this make sense? So now notice this is different than if you picked up the 5-pound weight first and then lifted up the 4-pound weight. If you lifted up the 5-pound weight first and then the 4-pound, then what would the threshold be? Then it'd be 20%, Katie. Exactly true. There you go, Kelsey and Ariel. Exactly. So you need to be very careful with Weber's law. There has to be an initial stimulus, and the initial stimulus must be defined for you. That's why I made a point to talk about left, because if we were going the other way around, it would actually be quite different. Perfect. All right, so let us know if that does not make sense. Mostly you can do them. I don't think we have the math right there, but you would take the initial stimulus, of course, and the change is 1. So you take the change and divide it by the initial stimulus, which is where you get 1/4. So it's 1 pound heavier. The initial was 4, which brings us to that 1/4 threshold. So then, what is the difference here? What would the difference be between these two weights? So still 1 pound, but what's your Weber's law difference, your perceptible difference? This wouldn't be the just noticeable difference, but because we already defined that threshold. Pretty close to it, Tadal. It's 1/9 exactly. So notice the difference in weight once again is equal to 1 pound, but the initial weight was 9, so it would be equal to 1/9. 1/9 exactly true, which is around 10%. It's a little bit more than 10%, but pretty darn close to 10%. Cool, exactly. So this person, though, since we know his threshold is 1/4, and since 1/9 is less than 1/4, we would say this is below the threshold. So we can't tell the difference between these two weights. Conversely, what about this one? Would he be able to perceive the difference, or would he not be able to perceive the difference in these two weights? And in all these, once again, we're saying the left hand is the initial. He's lifting up with his left hand first in every example because, of course, it changes depending what the initial stimulus is. It's back to 25%, exactly true. So once again, the difference in weight is 10 pounds. The initial stimulus was 40 pounds, so you just take 10 divided by 40, which brings you right back to 1/4. So notice he would think that the difference in these two weights is basically just as perceptible as the difference in those two weights. So the key is that it is a fraction. It's a proportion. It doesn't depend on the absolute difference. We can't just tell 1 pound, because if you lift 100 pounds and lift 101, you're not going to be able to tell that the 101 is heavier than the 100. But if you lift a 1 pound weight and then a 2 pound weight, it's going to be very, very blatant that the 2 pound weight is heavier than the 1 pound weight. And once again, remember, you're always dividing by the initial. I just wanted to make a point you are aware that it makes a big difference what your initial stimulus is. Okay. All right, so we had a couple questions coming through Q&A. Let us know if you have any more. But that's the general idea about Weber's law and how it'll work. It's different for everything. Like you'll have a different thing for, like photoreceptors, right, for weight, for auditory. Like a trained musician will have a much smaller, just noticeable difference than someone who's not a trained musician. Most of us can't tell the difference between like a 400 hertz and 401 hertz. But some people can. They have to be very, very good, but some people can. All right, so as we said, it's based on internal and external factors. The internal factors, of course, is how trained we are. External factors is the way it's presented. So is there a B on this page? Well, yeah, clearly there's a B. It's right here. So now here's my question for you. Let me know if you notice a B on the next page, and tell me in chat if you do notice it and when there is. There's actually only one B right now, just that one. Notice the rest of them are all. Oh, that's true. That's a good point. There's a B up there, Livingston. I see what you're doing there. All right, so there we go. So how about this? Let me know if you see a difference in B's. Is there a B there? You guys are pretty quick. There is a B. Where is the B? There is. There actually is. It's much harder to notice, though. It's in the bottom right, exactly. So notice the B is actually right there. But notice now internally we have the same perception. We've seen B's and P's for the past 20 or whatever years it's been. But of course this one, the external factor makes it much easier to identify. For this one, the external factor makes it quite a bit harder to identify. So that's the differences between internal and external. We all have very comfortable command of the English letters, I'm sure, at this point. Exactly the way they're presented makes a big difference. All right. So adaptation, on the other hand, so this is a change in threshold due to environmental change. This would be something, for example, if you get into a bathtub or a hot spring, at first it could be uncomfortably hot, but as you sit in it for a little while, it starts to feel more comfortable, more comfortable, more comfortable, until you don't even really notice that it's exceptionally hot anymore. Same thing when you go swimming in the ocean, right? Well, especially if you swim in California's ocean, because it's really cold there. But if you go in at first, it feels friggin' cold. You're like, oh my god, this is so freezing. But then within maybe 10-15 minutes, you can swim around, play, and you don't really notice that it's as cold as it was when you first got in. So it's basically adaptation. You're adapting to the differences. So at first it's super hot, and then eventually it's pleasantly warm. Same thing works for cold water as well. It also works due to, of course, time. So notice at first when you walk into a bathroom, you won't see a thing if the lights are off, but as you wait, it starts to get a little bit easier to see as your eyes start to adjust, as your pupils start to dilate. We'll get into eyes and all that stuff, and you start to take in more light than you do eventually are able to see. Cool. All right, so adaptation, just environmental change as it goes. And it's a good point, Ariel. If you're swimming in the ocean, you can never really complain. It's usually a good day if you get to go swimming. All right, so now I'm going to move this poll off right here, and I'll give you guys this very first question. We're going to go through several questions today. And after each major section, we'll give you another poll. So please throw your answers into this poll. Once we have majority answering, we'll talk about it. All right. Cool. All right, yeah, that's most of us. Nice work. We've got 96% of you saying answer choice D. I completely agree. Keyword in the answer stem was pain, unpleasant feedback, stubbed his big toe. Exactly true. And we talked about all the other receptors already. Hair, primarily we talk about, at least for the MCAT, the ones in the ears, photoreceptors, rods and cones in the eye, and then osmoreceptors is the concentration of the blood in the hypothalamus primarily. There's other places too, but that's the big, for humans, that's the big one. All right, so nociceptor, that means pain. Good work. All right, let's move on to the next learning goal. So this is a couple special senses. We'll talk about them, and we will start off with the most beautiful structure, by several people say, of the human form, which is, of course, the eyeball and how it works. So this is your eyeball. Of course, that's the front of the eye. That's the back of the eye. The cornea is, of course, where light converges. And so the cool thing is, I know that we say that light converges if you have a converging lens for the lens of the eye, but the cornea also causes light to converge. So you actually have two different lenses there that causes light to converge. And so, of course, the pupil is the part right there. It's what lets light in. It either constricts when it gets very light, or it dilates, of course, when it's in the dark. Let's tie in sympathetic, parasympathetic for this, though. If you had strong parasympathetic activation, which would you get? Constriction or dilation? Constriction, exactly true. So when you have extreme parasympathetic activation, the pupil will constrict, and when it's very sympathetic, the pupil dilates. So this is actually one of the warning signs that you need when you're working in the emergency room. If a patient comes in with pinpoint pupils, what would that probably, not probably, but possibly show? What kind of drug? What kind of drug? Shock, if they were actually like panic mode, it would actually be the opposite. They'd have a big blown out pupil. You got it. It would be a specific type of drug. It would most likely be an opiate. Exactly. If they're on some kind of an opiate, like a depressant, like if it's a heroin overdose or something like that, they're going to come in with pinpoint pupils. Whereas if they have some kind of a stimulant, then their eyes will get all dilated. So any kind of stimulant drugs would do that, whereas opiate drugs would cause pinpoint pupils. Like MDMA, exactly, perfect example, causes major pupil dilation. Any kind of stimulant would do that. And so it's certain things you can do, of course, to identify what's going on with the patient. It helps you figure out what you need to do. And in fact, if someone did come in an opiate overdose, do you know what you give them? Oh yeah, and you can totally. And there's other things you can do it, like pesticide poisonings can actually cause pinpoint pupils as well, and there's some other things, but you have to take in a lot of other information to decide if it is a drug overdose or not. But yeah, there's an actual specific drug you give patients that have OD'd on opiates. Blocks all of their dopamine receptors instantly and pisses them off dramatically. Get really upset about it. Narcan's actually the name of it. There we go, Amanda knows it. Narcan, yeah. So Narcan basically instantly brings them out of their high and makes them furious. Furious, because you just, of course, woke them up. So you have to be careful. Time down before you give it to them. Very important rule. I believe it's an injection. I've never given it before, but I'm pretty sure it's an injection. Cool, nice work. So the reason that we're tying in things like medicine and sympathetic, parasympathetic nervous system is this is what the MCAT does. It's not just going to talk about light and dark in the pupil. It's going to tie together multiple concepts, and so we always try to do the same in these classes as well. Cool. All right, so there's that. That's the iris, of course. So now the iris, that's just the colored part of your eye, right? So the colored part, of course, is the iris. The pupil is the dark part in the middle. Remember, the pupil is the only part that actually lets come through. Cool. As we said, the cornea is very clear. It's like a dome-like window as everything comes through, passes through. Also, let's talk about the different chambers. I'll just open all this stuff up so we can kind of see everything as we go through. So notice that iris itself has contractile muscles. We have two different ones. One is the contractor. One is the opener. One causes contraction, of course. The other one causes relaxation and pulls it open. Whenever you contract it, it lets less light in. When you open it up, it lets more light in. Also relates to sympathetic, because if you have your sympathetic nervous system activated, fight or flight mode's in, so your pupil dilates really big so you can let more light in, so you can perceive what's going on, understand your entire surroundings, and then potentially have to either fight or not or run away. I like Chuck's got a really good way to break that down. I'm going to take that from you and start using that myself. Perfect. All right, so cornea focuses, lens focuses. Now, where do you want that light to actually focus? Where ideally would the light focus? Right on the retina, exactly true. So the retina, of course, is the back of the eye right there. What's the fovea, though? Ariel brought it up. That's a big one. Exactly. You got it, Jennifer. It is. So that's actually the blind spot's not the fovea. That's the optic nerve. We'll do an experiment with that in a second, though, Min. But it is where all the cones are. You've got a very high concentration of cones in the fovea. And what do cones see? And we'll get more specific in a second, but what do cones see? They see color, exactly. So if the fovea is where you are directly focusing, and if you look right at a point, that means that the light is actually focused on your fovea right there. So have you ever noticed that if you go outside at night and try to see something, and if you look directly at it, you actually can't see it very well? The easier way to see it is actually not look at it, but look somewhere else and see it in your peripheral vision. That's because cones see color. And if you're outside at night, there's no colors to see, so you don't want the light focusing on your fovea. You want it focusing somewhere else on your eye, which is where, of course, more rods are. Because what do rods find? It's funny, but it's totally true. Totally true. You can't look at things at night. You're not going to be able to see it. It's because of your fovea. Rods primarily just see white and black, exactly. So at nighttime, it's easier to use your rods just to see the general outline, the white and black. But if you stare at something directly, you're not going to see a thing unless there's actual colors to perceive. So it's definitely good to know about the fovea. And we'll talk about that blind spot in a second. That's another good one. All right, so that's the focal point. That's where the light should all be focusing directly, because if you're looking right at something, we're usually doing a very fine analysis and looking at it very closely. The rods, of course, it's more your peripheral vision, seeing our backside. Cool, and that's your full retina. We'll get real specific into the retina. We'll talk about rods, cones, all the different types of cells, and get real specific with all that stuff. But it is your image detector. It's where we convert our photostimulus or light or photons into electrical stimulus to actually travel to the brain. Cool, nice. All right, so that's your optic nerve. Now, the interesting thing to know about the optic nerve is what do you actually not have on the optic nerve? Where the optic nerve is. Notice it's basically the retina is all there, but you have this one spot that there's your optic nerve, which means there are no rods or cones, which means you can't actually see if the light is focusing on your optic nerve itself. And so what I thought would be kind of a fun thing to do, and I made this up a little bit before class, is I want to do a little experiment. So you guys might have done this before, depending on if you had a class where you actually talked about all of these things. But let's do a little experiment where you can actually find your own optic nerve. Cool, this is pretty fun. I like this one as well. So what I recommend doing is you can do either eye, but we'll cover up our right eye. Actually, cover the left eye first. That might be easier. There we go. So cover up your left eye. Put your right eye basically in front of the X, and now slowly move your eye towards the S, focusing on the X. You'll notice at a very specific point in your peripheral vision, the O disappears. So start back here, look at the eye, get X, get closer, 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 and then there'll be this point that the O straight up disappears. You can't see it. You literally only see the white background. You guys see it? Pretty cool, right? So what's happening at that point, the light where that O is directly where your optic nerve is, which means it's literally a blind spot. You can't see it. If you want to get even cooler, what I recommend is do the same thing. So cover up your left eye, focus on, find that blind spot. As soon as you're on the blind spot, then lift your eye, close your other eye, so you're flipping around, and then look at the O. And you'll notice that when you focus your other eye on the O, as long as you don't move your head, the X disappears. So that would be, of course, the blind spot on the other eye. Isn't that pretty neat? Science is so cool sometimes. There's so many experiments you can do. And so it's super easy to do. All you need to do is a piece of paper. You can do 2 X's, you can do an O, whatever it is. But you can make these yourself anytime that you want to. All right, so we won't do any more of this, but it's good to know about it. And this is a nice way for you guys to actually remember about the optic nerve and that it is a blind spot since we're talking anatomy and structure. So cool stuff. All right, so there's our little experiment for the day. We can do more in the future, but that'll be it for now. All right, so that's the structure of the eye. Let's kind of dial in and get a bit more specific about the actual cells and the structure and the kind of like the big picture here. So rods and cones, as we said, these are the light-absorbing cells of the retina. So you have 2 main types, of course, well, rods and cones. We have a couple different types of cones, though. So they're the things that are responsible for transduction of light energy into electrical signals, right? Our brain only perceives electrical signals, so whether you have chemical signals or light photoreceptors or whatever it is, it needs to be first turned into, of course, axons and electrical signals going down the brain. All right, so first, of course, we have rods. That's the front of the eye. Basically, your synapse, that's where, of course, your photons will hit that point. Notice the back of the eye, that's where your actual rods are right there. So you have the nucleus of the cell, all the mitochondria supporting all the energy, and of course the very back, that's where the rhodopsin is. So the rhodopsin is the thing that does what we need. It's the perception of the different either types of lights or colors or whatever it is that we're referring to. So do know the word rhodopsin. It's very important. And that's of course where, of course, the term rods comes from rhodopsin. All right, so that's a rod. Notice it does actually look like a rod. Mostly be aware the structure of it, it is in fact behind, whereas the light's coming in from the front. So that's a little bit funky for a couple people. All right, so now do be aware for rods, there is just a single rhodopsin pigment. There is only one, which is why we can only perceive black and white. You have pretty low sensitivity to detail, but it does allow us to see at night. Cool. Oh, that's the actual, that'd be the retina, I believe. So it'd be like kind of like the line right there, or they'd all be attached to that right there. Let me know if that's not it. The other teachers can let me know if it's slightly different. But yeah, it's like they're all kind of attached in that midpoint right there. So they'd all be lined up vertically. So it'd be the membrane, I guess you could say. All right, so remember, there is no color perception at all between the two, at least for rods. Cones are the cool ones. So, and also remember, this is just big picture. You don't need to memorize the structure of these things. The main thing you need for test day is know rods are black and white. There's only a single pigment for your rhodopsin, and they're not sensitive to detail, right? Just basically, you know, color. Not color, exactly. Black and white, that's the main thing. It's where the optic nerve is, Bellany. It's where your optic nerve is. We'll get back to optic nerve in a little bit, too. All right, so now cones, on the other hand, they, of course, can see color. So once again, you have the synapse up front, you got the nucleus, you got the mitochondria. The membrane shelves are lined with rhodopsin for cones, right? You've got 3 different types of rhodopsin pigments for cones. So now, thing to be aware of, these are sensitive to color, they're sensitive to detail, they work the best in bright light. The concentration of these are at the fovea, that's the thing we talked about before. So at your fovea, you do have a high concentration of your cones, and so there we go. So if you look very closely at something, you're going to have lots of cones. So now, interesting thing to talk about cones, you might have heard of people, of course, having color blindness before. There's a couple different types of color blindness. Generally, it's red-green color blindness. But one of the types of color blindness, we only have 2 cones instead of 3 cones. And so the problem is we actually have 3 different types of cones. There's long, there's medium, and there's short. And it's that, how about this? If we can't really perceive the color red so well, which cone do you think we're missing? The long wavelength, the medium wavelength, or the short wavelength? Let's connect the dots. It's the long wavelength, right? Because long wavelength means small frequency, and red is the lowest energy, thus the lowest frequency, thus the highest wavelength. And so some people, one of the types of red-green color blindness, it does mean that you actually are completely missing the long wavelength cones, or they just are faulty or not working correctly. Even cooler, do you know that some humans, primarily women, actually have 4 different cones, not 3? There's still a lot of research figuring this out and how it works, but for real, some people actually have 4 cones, which means they are even better at perceiving differences in color. It's pretty cool. And so, you know, like when you hear people like, oh, no, that's not white, that's mother of pearl. No, they're better at defining differences between. We're still, like, the problem is, you can't really, if you saw extra colors, you'd be seeing ultraviolet. The problem is as light enters the eye, the ultraviolet's actually filtered out, I believe, by the cornea. And so ultraviolet doesn't make it all the way back to the retina. And so you can't really see the other colors because they're basically filtered out. But they're just better at basically perceiving differences. And there's actually some organisms that have up to like 12 cones. If you haven't seen a mantis shrimp before, I recommend looking up mantis shrimps. They have like 12 cones. They are the most colorful creatures in the world. And so they can basically perceive all the differences between each other because they're really, 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 really good cone systems. They have very good color perception. It's called a mantis shrimp. It's a mantis shrimp is the name of that animal. Actually, if you like the oatmeal has a really cool article about them. Yeah, exactly. So know about your rods and know about your cones. And the more relationships and stories you have, the better it will be to remember them. So. All right, so now we're going to just do the big picture here. There's a lot we can talk about, but I do want to talk about the other types of cells that talk to these rods and cones and give us the entire picture of light coming in. So now, as we said before, remember that light coming in actually travels all the way back and then hits the rods and cones behind everything else. So it's kind of funky in the structure, but it is, of course, deeper in the back of the retina, right? So there we go. That's the front of the retina, the back of the retina, and we said there's your border right there between. So now the rods and cones are in the very back. There's a cone cell. There's a bunch of rod cells. We already talked about those. Now the front of the retina, though, has different types of cells. Now we've got bipolar cells, and so we'll kind of get the big picture. But if you bipolar cells, you don't have a one-to-one relationship between rods and cones and bipolar cells. So as you have a whole bunch of rods and cones all converge together to bipolar cells. And so the more rods and cones you have connecting to a single bipolar cell, what happens to the distinction between colors? More or less distinction between? Less distinction, exactly, Katie, or everyone, you got it. So the idea is if you have a bunch of different rods and cones connecting to one bipolar cell, you're not as able to really differentiate between different colors or lines and all that other kind of stuff. Whereas if you have fewer, you have better distinction between. And so everything, of course, is going to converge to these bipolar cells. It is a direct input from your rods and cones, and the more rods, cones you have going to it, the less distinction you're going to have between. And so these synapse with the ganglion cells, those are the ones we have up front. The ganglion cells, these are the guys up front here. They're the ones that travel to the optic nerve. So that's your ganglions up there. They form the optic nerve eventually, which then travels all the way down to the brain. All right. Now, we have two other types of cells. We have horizontal cells and amacrine cells. These are a little bit different because they actually, well, between the two, they're kind of secondary messengers. They're not directly linking to the rods and cones. They're connecting to bipolar cells and helping with other kinds of distinction. Namely, they're going to receive a bunch of different info from a bunch of different types of retinal cells, the more the complicated players. They help us define edges. They help us define contrasts. It's the relationships between all of them. And so main thing you need to know, we've got bipolar cells. These bipolar cells do directly connect to the rods and cones. These other, not really helper cells, but the other types of retinal cells are horizontal and amacrine cells. They receive info from multiple different types of retinal cells, including the bipolar cells themselves. And they're the ones that detect, of course, edges, contrasts. Basically, it's inputting a whole bunch of information, helping us put the whole picture together. And then finally, the ganglion cells, as we said, are up front. They're the ones that converge together to form the optic nerve. So this is everything's going to eventually get that, and then it's going to travel all the way down and go to the brain. So mostly know your types of cells. Know bipolar directly connects to rods and cones. Amacrine, horizontal, they're intermediaries is a way to think of them. They connect to multiple different retinal cells. And then the ganglions are kind of like the last step as we get to the retinal nerve, or the optic nerve, excuse me, going down to the brain. All right, big picture, right? As we said, we're not going to get too specific. We could get really into the differences between, but we want, of course, the test day high-yield stuff. All right. So visual pathway, this is a pretty cool thing. So when we talk about the anatomical and physical connections between the eyes, the main thing that you need to know is the way that it gets to the brain. Now, I do recommend, if you've never seen this before, to actually either take a screenshot of this or get used to drawing it. But the idea is, notice that all of the light coming from this side of the field. So we could draw this as actually like a line straight down the middle. And so light coming in from the right visual field is all going to travel to, notice right here, actually let me make this smaller so it'll be easier to see. And it's going to travel to, of course, the left side of the brain. Same thing, as long as it's coming from the right visual field, regardless if it's the left eye or the right eye, it's going to travel to the left side of the brain. Conversely, any information coming from the left visual field, notice is going to go in, and it's going to go to the right side of the brain, regardless if it's coming in from the left eye or the right eye. So if you were to actually look straight forward, everything you see beyond your nose, like the nasal bridge right there, to the right, is being processed by the left side of your brain. Everything you see to the left is processed by the right side of your brain. It doesn't matter what eye you're talking about. It's not like right eye one side, left eye the other side. It's actually both eyes are putting information to both sides of the brain. What matters is the actual field of vision that you're looking at. So that's one thing that's actually a common misconception. It's not right eye, left eye. It's all about the actual visual field that you're looking at. And so right in the center, we have the nasal fibers. So notice that the nasal fibers, so the nasal fiber of the left eye, of course, is going to the right side of the brain. The nasal fiber of the right eye is going to the left side of the brain. But if you look at the outside fibers there, they're the temporal fibers, right? Temporal lobes in the back side of your head. Or you just think temples are right here as well, same word root. They're actually sticking to the same side of the brain. Notice that the temporal fiber on the left side does go to the left side of the brain, but it's because it's pulling information from the right visual field. So nasal fibers, you've got this switched. It's known as the optic chiasm. So the optic chiasm is where you do have those navel fibers crossing and going across the opposite sides of the brain. And so which not hemisphere, what's the name of it? Which lobe, I guess it's called lobe. Which lobe actually perceives visual information? The occipital lobe, exactly. I was like, is it called lobe? It is called lobe. But yeah, the occipital lobe, of course, the back part of your brain right there, it's the one that processes visual information. Temporal, of course, on the side. We got the frontal in the front, and parietal as well. We'll get into those in future classes. We don't get too much into the different brain lobes. All right, so, and of course, you've got your lateral geniculate nucleus. There's a mnemonic to be aware of because you've got your lateral geniculate nucleus and you've also got your medial geniculate nucleus. And so the lateral geniculate nucleus deals with light. So you've got an LL mnemonic between those two, so it perceives light coming in. Do you know what it's a portion of? It's actually part of a brain structure that perceives incoming information. It's an important part of the brain you do need to know. Thalamus, you got it, Ariel, exactly. It's part of the thalamus. And so thalamus basically is very much a relay station that perceives information coming in from outside portions and then directs it to where it needs to go. So exactly. And then it goes to the visual cortex, as we said, in the occipital lobe. Cool. All right, so as you said, right visual field goes to the left hemisphere, and left visual field goes to the right hemisphere, as we showed on this page already. So know that. Know it loops through the lateral geniculate nucleus, which is part of the thalamus. Then it goes to your lobe and actual processing in the back. All right, so this is what we pretty much already talked about. Just a couple more pieces of information to be aware of. We already talked about the thalamus, the lateral geniculate nucleus. After that, of course, we're going to cross through the middle. That's known as the superior colliculus. Once again, superior colliculus deals with vision. This is part of your midbrain. We'll talk about the inferior colliculus, which deals with hearing later on. That's also, but it's still part of your midbrain. So know your colliculi are part of the midbrain, and then the very back, as we see, is your visual cortex. So the superior, the midbrain structures. Actually, any guesses what midbrain structures would kind of like help with? Things as you get more midbrain located. Less processing, more what? More vital. You got it. It's more like senses. It's more perception. Like, for example, when your midbrain, like for your hearing, for example, if you hear a sound, the midbrain's gonna be like, oh, the sound's coming from that direction. It's more sensory, figuring out where it's coming from. It's more reptilian functions. You know, like any animal can hear where things are coming from. It's not really the processing. They're like, oh, that sounds like my brother singing this song. It's more sensation. You got it. More sensory input. You got it. So midbrain's more. It's more reptilian. You could think of it. More like perceiving where things are coming from and where they are. Cool. All right, so parallel processing, we talked about a couple different classes, but pretty much it's just talking about a bunch of different information all coming in and then combining it all together to figure out what it is. So in order to identify the animal in the picture, what are you going to do in a very structural sense of it? Things. To identify this animal, you would first say, oh, hey, look, this animal has spots, first of all. It's got orange fur. It looks like it's got that tear mark face under its thing, which means it's some kind of a cat. You got it. It's got the cat face. It's got the nose of a cat. It's a big cat. Namely, it's got those ears. It's a cheetah. You got it. And so then finally, once it gets to the visual cortex, gets processed, you're like, oh, yeah, that's a word for that. It's called a cheetah. And then we start thinking of lots of other things, like, oh, that's the fastest land animal on the planet, or, oh, they like to eat gazelle. And all the other background information that we know about cheetahs starts to come forth once it makes it to that cortex. Gets all bounced around all over the place. Also, we can use things like feature detection. So, for example, if you look at this crowd, you're like, oh my gosh, there's a bunch of people. But if you're looking for your friend, you want to think of a couple of things first. Like, what would you want to know about your friend most importantly if you're trying to find your friend in a crowd? You got it. What color are they wearing? What's their hair look like? What's their hat they're wearing? And so you'd say, okay, well, we know where they're wearing green. And so then notice as soon as I said green, do you notice people wearing green? Bunch of them, you're like, oh, look, there's a green person. There's something green right there. Looks like there's something green right there. And then you start, and there's a girl in green right there as well. You start picking up all the little individual pieces. And so it allows you to detect a specific feature depending on what it is that you're looking for. You don't need to take in the whole scene. You only need to perceive the things that are in green. That's called feature detection. So color, good example. Once again, we use our cones to get color. Shape, of course, is your paravellular cells. We haven't gotten into this too much yet. We get way more into it in the review notes, but those are the ones that help us define shape. It's the relationships between. All right, motions, the other ones, those are magnocellular cells. So other things you do want to be aware of, shape is paravellular, motion is magnocellular. The way that I always remember this is just motion and magno. It's both MM root. I don't have a good mnemonic for shape and paravellular. Maybe one of the other teachers does, but I haven't figured out one yet. I'm sure in the next couple months we'll figure out something for those. But there's a bunch of little things we can do, kind of like lateral for light, M for music. There we go! Parallelogram's a shape. I dig it, Jennifer. We're going to do that. Parallelogram, shape. Magno, motion. Perfect. I like it. So a couple of things to be aware of. Let's do some questions, though. We don't have too much time left, and we want to make sure we do get to a couple more main points, namely to hearing. So throw your answers into this poll, and then we will discuss further. All right. There's a number one mistake people make in these types of questions. Any idea what the number one mistake or trap people would fall into there? There's one word in the question stem that matters more than everything else. You got it. Incorrect. So we're looking for something that is not true, which means 3 of these are correct, and 1 of them, of course, is incorrect. Perfect. So now there's also a fun thing you could do here. Notice if you look at B, C, and D, they're very similar, so we're probably going to have 2 opposites and one that's, basically saying the same thing. And it looks like we are definitely rocking this. We have 75% saying answer choice C. Notice all fibers from the left visual field project to the left side of the brain. Nope. Of course, if you know that the right visual field is projecting to the left side of the brain and the left visual field goes to the right side of the brain, then of course you can eliminate both of those because you can't have 2 correct answers, or in this case 2 incorrect answers. So the answer does have to be C, as most of you were saying. So even if you didn't know, you can use the structure of the answer choices to eliminate and find a good guess. All right, that's a big part of it. All right, let's get into the ear. So now hearing is another cool thing. We discussed some of it already, but we'll get into the big picture stuff. So the external ear to the tympanic membrane is the external. Anything outside the tympanic membrane is known as the external ear. What's the first part of your ear called? The part that people put holes in sometimes? That's pinna, exactly true. So it's the pinna, sometimes called the auricle as well, but pinna is the most common word that I've seen it referred to in most of the writings that you'll see. So that's, of course, just the cartilaginous outside portion of your ear. The earlobe, of course, at the very bottom, and then that's your ear canal, or the external auditory canal, to use the fancy word for it, coming in. So this is all external ear. Even the external auditory canal is still outer ear. You don't get into inner ear until you get past the tympanic membrane. So once you get past the tympanic membrane, that's when you get into the inner ear. So sound waves travel in. Notice that the structure of the pinna lets you hear sounds more loudly because they all get converged. In fact, if you've ever done this before, but you can actually loop your hand around your ear when someone's talking across the room, and you can actually hear them a lot more clearly if you do increase the surface area there like that to let more sound waves converge. It's kind of like the old men from the 1920s with the cone they would just stick in their ear and point it around. The original hearing aid. Just use your hand. Same idea. All right, so now the thing is, as all those sound waves converge into your ear, it causes the tympanic membrane to vibrate, just like a timpani drum vibrates, the exact same idea. So now, the tympanic membrane now has a very special bone stuck to it, and we got 3 of them specifically. You've got the hammer, the anvil, and the stirrup. The hammer, of course, known as the malleus, like mallet, malice, hammer, same idea, is actually stuck to the tympanic membrane. So when the tympanic membrane vibrates, it causes the malleus to also vibrate. The malleus is connected to the anvil, which is known as the incus, once again, anvil, incus, similar word root, which then is connected to the stapes, which is the stirrup. Now the stapes is connected to the oval window. So once we get to the oval window, that's when we change from the middle ear to the inner ear. And what do you call this canal that's going down the middle ear all the way towards the mouth? Yeah, and Chuck's got some good information in there, too, for you. Yeah, the eustachian tube. That's like when you plug your nose and you blow, that's forcing air up your eustachian tube into your middle ear, which is why you feel your ears pop and expand. Bingo, pressure regulation. Totally correct. All right, so as we said, malleus is fixed to the tympanic membrane, acts on the incus, incus acts on the stapes, stapes rests on that oval window. So what we're basically doing is converting vibrations from the tympanic membrane all the way to the oval window through some actual ossification, some bones, right? Ossa, right, word root for bone. All right, and that's your eustachian tube, as we said, which is all about pressure. You guys already called that out. All right, so now the inner ear, this is where things get really interesting. It starts at the oval window, and it goes all the way to the auditory nerve. Now, this image is also in your review notes. It's in the Psych Soc book, so it has everything labeled there, so don't feel like you have to write everything down here. We're just going to talk big picture. So we've got semicircular canals up top. Semicircular canals deal with rotational motion. So rotational acceleration, not linear. Be very careful. It's a common misconception. The parts that deal with, there we go, actual linear acceleration are in the vestibule. So the vestibule, we talk about vestibular senses, right? Same idea. Those are the ones that are sensitive to linear acceleration, speeding up, slowing down. This is also why, have anyone ever gotten an ear infection and then noticed that they can't stand up straight and they don't really know where they are and they get super confused all of the time? Potentially, maybe not. It's like one of the possible things, if you get an ear infection, it actually gets into the inner ear, and all this stuff can get inflamed, and oh my gosh, that's when things get very complicated. You can't really perceive where you are, and it gets really complicated. Balance gets completely messed up. All right, so balance, orientation, and space is what that's all about. Now, how does it actually know whether you're accelerating or not? That's the kind of weird part. How do you know that you're accelerating because you have these things in your ears? So we got fluids, and in those fluids, we actually have fluids move, and the fluids move something. So we do have a lot of fluid in here. There we go, exactly. The hair cells inside of it. So due to the hair cells and the momentum of those hair cells, that's what, through a chain of reactions, basically lets you know that there is motion and lets you know that you're moving and have acceleration. Nice, you guys got it, totally true. All right, so they're sensitive to movement. You can perceive when they're going. So vestibular and auditory input. Notice that your cochlea that we have down here and all of your semicircular canals and vestibule go to the same place. They all go up that inner ear. Sorry, the actual auditory nerve, excuse me. From the inner ear to the auditory nerve. There we go. All right, cochlea is pretty cool. Cochlea is the actual hearing apparatus. Once again, it's hair cells that perform the signal transduction. So it's through the vibration of hair cells that turns it into, of course, nerve impulses, which then travel all the way up the ear, the hearing, or the nerve as well. Yeah, exactly, the nerve. And so we've got fluid separated by membranes. We've got two different types. We've got perilymph, and we've got, of course, endolymph. So the thing is, the perilymph both cushions the structures and allows the sound wave vibrations to travel through it. The endolymph is basically like a nutrient-rich fluid that all the hairs are bathed in. So it's really good. It has all the nutrition, all the stuff that they will need to survive. But the sound goes through perilymph. You need to know that one. That is important. That's where the sound waves transmit through. Now to look a little bit closer, to kind of zoom into the cochlea, now the structure of this is the actual perilymph on the outside. That's the endolymph on the inside. And what's the name of this organ with all the hair cells on it? That's the organ of Corti, Bellany. Exactly true. So the organ of Corti, that's the thing with all of those hair cells on it. And notice that it's resting on the basilar membrane. I actually don't know about tinnitus. We'll see. One of the teachers will. That's a good question, though. But exactly. So resting on the basilar membrane, you've got the organ of Corti. And so the cool thing about the basilar membrane is that it's actually structured in a way like this, where this would be the oval window right here. And so what types of sounds would you hear if it was a narrow portion of the basilar membrane? High frequency or low frequency? This would be your apex up here. So it's kind of like the center of the clamshell. Exactly. So down here you'll have high frequency because notice it's smaller, right? It's stiffer, it vibrates at a higher speed, right? So it's, of course, higher frequency sounds. Whereas in the apex, which would be the center of that snail shell, that's where you hear low frequency sounds, all the way down there. Cool. So that's the basilar membrane. It's, of course, vibrates, and that's, as we said, so, of course, by the oval window, you hear high frequencies. As you get closer and closer to the apex, it's lower and lower and lower frequencies. And since that near the oval window, it's very stiff, that's like as you get older and older and older, it gets stiffer and stiffer, and eventually stops vibrating, and eventually you can't even hear high-frequency sound. So like old folks really struggle to hear little kids talk because near their oval window, their basilar membranes basically become so stiff that it can no longer vibrate. All right, so those are your stereocilia of the hair cells. Of course, through them moving, it opens up ion channels, which then transducts and gives you electrical signals from the motion of the hair cells themselves. All right, so notice these are mechanoreceptors, as we talked about before, because they're receiving movement. Right, so they are, of course, mechanoreceptors. Main things know basilar membrane, organ of Corti, and of course know how, the structure of and how sound waves traveled on it. Cool, and of course from there it goes to the auditory nerve. So basically the big takeaway here is basilar membrane vibrates, causes the hair cells to depolarize due to shear. Shear means basically bending to the side from the vibration, which then of course causes an electrical signal to get sent up, down, all the way through the nerves to the brain to perceive it. So that's your pathway. Now, the main thing that I want to talk about with the pathway is how, of course, it's different from what we talked about before. So the auditory nerve is going to go to your superior olive. So now, we won't get too much into the superior olive, but superior olive is actually a brainstem structure, which is more, like actual life and death kinds of things. You know what I mean? More like, brainstem, very reptilian brain. After that, we get into our inferior colliculus. As we said, this is a midbrain structure. It's all about like localizing, seeing where sounds are coming from, perceiving where it's at. Once you get past that, then you get to the medial geniculate nucleus, which once again, medial geniculate nucleus would be part of what? Medial geniculate nucleus is part of your thalamus, once again, exactly. So remember, also the medial geniculate nucleus begins with the letter M, just like music begins with the letter M, whereas the lateral geniculate nucleus, of course, deals more with your light perception, all that. Yeah, more like reflexes, you got it, Ariel. Like kind of like, startling response and things like that, and moving out of the way of things coming at you. It's the more reptilian brain, not really processing. Cool. Gotcha. Totally true. And then after that, we go to the cortex. So, of course, temporal lobe, right next to the ears by the temple. That's where sound is perceived, and we go through the whole processing fact as well. So main difference, I want you to know medial versus lateral. Of course, we want to relate the different things together, so we know, of course, how they're the same and how they are different. So let's do this once again. Throw your answers into the poll. This will be our second to last thing. We'll just do a quick summary after this. Those are our two big senses. Those are the main senses you need for the MCAT, the vision and the hearing. I apologize for that. I muted the microphone. I was talking to the teacher secretly right there. But so there we go. So the last thing that the only thing we didn't really cover today was the olfaction and the smelling sense. Olfaction is smell. So the one thing I do want to tell you about smell is I just want to talk about how smell and taste are a little bit different. So the thing that you need to know about smell is that it's chemoreceptors, right? It's specific cells that are perceiving specific things. And when smells go in there, they go through the nasal cavity, activate the olfactory epithelium, which goes to the olfactory bulb. Now, notice, this is the only smell, the only sense that's not getting routed through a very specific structure. What did everything else have to go through that smell's not going through? The thalamus, you got it. So everything else goes through the thalamus. Smell's the one thing that goes straight to processing, which is why they often say that if you smell something, you know how it comes like really quick right back to like memories, and you smell something like, oh my god, that's that person that I knew this one time, or this memory from back in the day. One of the theories on why smell has that super strong sense is because it goes straight to the cortex. You don't have to process it through the thalamus. Even when you look at taste, taste is the same thing. It's still chemoreceptors. You've got the different types of taste that we all know, sour, salty, umami, bitterness, and sweetness, but they still have to route through the thalamus. It goes from the taste buds to the brainstem, through the thalamus, and then finally to the cortex for processing. And so that's kind of the big difference with the last two senses that I mostly wanted to do. Olfaction is the only one. It goes right to the brain. No thalamus there at all. All right. Thanks for coming today. I hope you guys learned a whole bunch about those main senses that you need to know for the MCAT. Let us know if you have any questions. We're going to hang out in the Q&A, but we're going to, of course, cut down the public chat right now as we jump to our final slide. So yeah, let us know. We got some handouts for you as well. And keep coming back. We've got lots of these happening all the time. Fun times, team. All right. See you guys soon. Have a good rest of your evening.